Illinois. So I'm ABC, and I guess I'd first like to welcome you to 2011, the Ecology and Evolution Seminar Series. And it is my distinct pleasure and indeed honor to be introducing you to our first speaker this year. Is also a speaker sponsored by the STORR Major Issues in Modern Biology lecture series. So our first speaker is Dr. Gene Robinson, and Gene is I think we could safely say he is sort of the ultimate integrative biologist. So as you'll see, his work spans from modern molecular genetics and genomics through physiology, neuroendocrinology, behavior of individuals, up through the behavior and function and ecology evolution of colonies of social insects. And because I think his work is so integrative, it turns out that at the University of Illinois, he has an amazingly diverse series of appointments. So I thought I'd read that out to you. And this won't take too long, but it will actually take a little while. So Gene, and you know, the fact that he studies genetics and his name is Gene, you know, that's something, I don't know. But in any case, Gene he's a professor in the Department of Entomology. He's also director of the University of Illinois Bee Research Facility, director of the Neuroscience Program, leader of the Neural and Behavioral Plasticity theme at the Institute of Genomic Biology. He also has joint appointments or affiliate appointments in the Illinois Natural History Survey, the Department of Cell and Developmental Biology, the Department of Animal Biology, the Center of Economic Entomology, and the Department of Political Science. So we can ask him later about the political science in particular, I guess. But also in recognition and fitting the quality of his research, he has won many, many major awards, including, and this could also take a little while, but I guess I'll do the brief version, that he has won the Burroughs Wellcome Innovation Award in Functional Genomics, the Founders Memorial Award from the Entomological Society of America, the Fulbright Senior Research Fellowship, Guggenheim Fellowship Fellow, Animal Behavior Society Fellow, American Academy of Sciences, Arts and Sciences Fellow. He's a highly cited researcher and a member of the National Academy. And I will finally also just note that I saw him give the major opening plenary talk at a conference this summer, and it was spectacular. And so we are in for a treat. So time for me to shut up and bring out Gene Robinson.
Well, thank you very much for that really kind introduction. I have a lot to live up to right now, so we'll get started. I just wanted to say what a pleasure it's been to be back to Davis. I have many dear friends and colleagues here, and met some new folks today, and so I had a really delightful time, and I'm just really glad to be here, kick off the new year with you. What I'd like to do is talk with you today about how we're using a variety of techniques, primarily in genomics, to address issues related to the evolution of social life in insects. So to begin with, I think we would all agree that social evolution has given rise to some of the most amazing traits that we know of in biology, traits that in many ways have changed life on this planet, some for better and some for worse. Language, agriculture, and warfare being among these kinds of traits. You're familiar with their occurrence in one set of species, of course, humans, but I wanted to dwell on their occurrence in the social insects. So we have language, an example of symbolic language, the honeybee, which we'll be talking about later, agriculture, the famous leafcutter, ants, and then warfare, the honeypot ants of the deserts of the Southwest that engage in warfare, and other ant species as well. So we have these interesting traits, language, agriculture, warfare, that clearly are the result of social evolution. And what's, I think, quite interesting about them is their unique phylogenetic pattern or distribution. So we have their occurrence in the social insects, and then, of course, in us. The last common ancestor, a marine flatworm living some 600 million years ago, very primitive nervous system, as indicated here. No social life, as far as we know. leading to some very profound questions about the role of conservation and convergence in the evolution of social life. So in the context of the insects, the most extreme manifestation of social life is what we call eusociality. It has, at present, 3 defining characteristics. There's an overlap of adult generations. There's a big difference in reproductive output between individuals. Some individuals reproduce and some don't. And then there is cooperative brood care, individuals cooperating to take care of the babies. These are the 3 defining traits of eusociality that make possible these spectacular traits that I was talking about before. So we have then first to confront the question of the evolution of eusociality. In many cases, once eusociality has been reached, once that threshold has been crossed, there's then a division of labor among the workers, among the individuals that do not engage in reproduction. Those individuals, as most of you know, in the case of the hymenopteran societies, the ants, bees, and the wasps, are called queens. And then the other females that do all the work related to colony growth and development are called the workers. So there's a division of labor among workers, and we'll be talking about that. So that when we try to understand and use genomics to understand the evolution of insect social life, we're trying to understand the evolution of eusociality and the evolution of division of labor. So I'm going to be addressing 3 areas, the 2 that I just talked about, the evolution of social life, by talking about some studies we've done in wasps and bees, in this case bumblebees and some other bees. Then the evolution of social behavior, specifically the evolution of division of labor in honeybees. And then the last topic, once a division of labor is in place, there can be adaptation of division of labor, changes in the tuning of the behavior, if you will, and I'll use as an example our work on aggression in honeybees. So when one thinks about the evolution of eusociality, the traditional approach has been to identify the evolutionary forces, such as group selection, kin selection, and so forth. I won't be talking about that today at all. Instead, I'll be approaching these evolutionary questions by a focus on the question at the bottom here, which is what kind of biological processes have been involved in these evolutionary processes, or how can we use genomics to make some inferences about the kind of biological processes that have been involved in the evolution of insect social life. And just to give you the tip-off, these are the kinds of insights that we've gained so far, hence the title, if you recall the title of my talk. So we have insights that implicate feeding, nutrition, metabolism, and reward as major players in the evolution of insect society. So I will spend the rest of the time talking about the evidence that supports this conclusion. So here's an outline of my talk. So for the first part, the evolution of social life and the evolution of eusociality, I'll be talking about the evolutionary transition from maternal care to sibling care in Polistes, paper wasps, and bumblebees. Then I'll be talking about a study on molecular evolution to find signatures of selection, that is, identify genes that show signs that they have undergone some sort of evolutionary process related to the evolution of eusociality. In part 2, the evolution of social behavior, which, as I said, is dealing with the evolution of division of labor. I'll be talking about hormone signaling and especially insulin signaling, juvenile hormone, and aspects of that. And then talk about the dance language and our work on cocaine. And the tip-off there is cocaine makes bees dance more. So you might want to stick around for that. And then finally, the third part of the talk on adaptation of social behavior, we'll be talking about pheromone regulation and aggression in killer bees. Okay, so let's begin. With the evolution of eusociality, there was a very insightful analysis that was performed a number of years ago by Mary Jane West Eberhard, in which she pointed out that maternal behavior can actually does consist of reproductive care and brood care. So reproductive behavior, that is laying eggs, having babies, and then the care of that offspring. And that's what we think of as maternal behavior. It encompasses both of those. That's the classic parent-offspring bond. And what she theorized was that these have become unlinked in eusocial evolution. so that reproduction is the province of the queen and care is the province of the worker. I'll just add parenthetically that it's interesting that the substrate maternal care has been unlinked because there are related theories having to do with the evolution of romantic attachments and monogamy in humans and other mammalian societies having to do with the redirection of that sort of behavior, maternal behavior. Happy to talk about that stuff later. So the idea then was that these became unlinked, and this was Mary Jane West Eberhard's idea. Linksvaer and Wade, about 30 years later, sort of modernized that approach in the context of Evo-Devo and hypothesized that you could think about this unlinking in terms of changes in gene expression, compartmentalization of gene expression, temporal changes in gene expression, culminating in sets of genes that are expressed for reproductive care in the queens and genes that are expressed for care behavior, brood care behavior, in the workers, then becoming, of course, not offspring care, but sibling care, caring for their younger sisters primarily, because the Hymenopteran societies are primarily female societies. So this was the idea. Linksvaer and Wade sort of, as I said, put it in a contemporary frame and gave us then the opportunity to begin to test this idea. So the idea then, we can see differences in gene expression that are associated with maternal care, or rather the splitting up of maternal care into reproductive and care modules, if you will. The primitively eusocial species are ideal for this because what you have is, yes, you have a queen, and yes, you have a worker, but before that, you have basically a single mom, a foundress, that does everything at the beginning of the colony foundation phase. An individual that founds the colony, establishes the colony, lays the eggs, goes out and gets food, lays the eggs, does everything. After the first generation emerges, she kicks back, becomes a queen, and then the workers take over all the performance of the care. So you have this great plasticity within the species, and the foundress is engaged in a lot of worker-type behavior. So we needed, of course, resources, genomic resources, to test this idea in an efficient way. So we used next-gen transcriptome sequencing. We've created EST databases, thousands of ESTs for the species that I'll be talking about in both parts of this talk. And then we did gene expression analyses, both by microarray and qPCR. We also used some analyses that we did in honeybees, where we look at genes that are differentially expressed in different behavioral contexts, and we have our quote care, sibling care gene list, that is genes that are differentially expressed between bees that are engaged in brood care versus those that are engaged in other kinds of activities like foraging. So these are changes in, or differences in gene expression in the brains of the honeybee. By the way, this foundational work, pioneering work on nurse forager division of labor was done by a former postdoc of mine, Charlie Whitfield, who was an undergraduate here in Ken Burtis' lab right here at UC Davis. So we use that gene list as the basis for our care gene list. And then, as I said, we studied first a paper wasp, Polistes metricus, and then we used a bumblebee in more recent work, Bombus terrestris. So this was the work of former graduate student Amy Toth, now on the faculty at Iowa State University. So we took a set of genes that were identified as care genes from the honeybee brain transcriptome studies, and then we hypothesized, based on the Linksvaer and Wade extension of Mary Jane West Eberhard's ideas, that the foundress brain gene expression profile should be more similar to the worker profile, because the foundress was engaged in all of these worker-like activities. And as you can see, that's exactly what we find. So I'm just summarizing the data for you here, 2 multivariate statistical analyses. So here we have a linear discriminant analysis, Q for queen, G for gine, I won't talk about them, W for worker, F for foundress, and you can see a lot of overlap in the brain gene expression profiles of foundresses and workers. And another statistical analysis may be making this point a little more clearly for you, showing that the brain gene expression profiles of these 2 groups were more similar to each other than to the queen groups. This was a set of 32 hand-picked genes from the honeybee care list. It looks like those care genes might be special, as we might expect, because when we take a random set of genes from the Polistes brain transcriptome over here. This is now the result of a microarray study. When we take a similar number of genes, but now randomly selected from the list here, you can see no such connection between founders and worker. These are the old data that I just showed you, and these are the new data that do not show this overlap. So the idea seems to be supported by the transcriptome data for the specially selected care genes. And then the last piece of data to support this for today deals with a new study that we've done that basically asks the same sort of question, but for bumblebees, for Bombus terrestris. And what you see here are the care genes, that is the genes showing differential expression in bumblebees. We had 1,180 differentially expressed genes between reproductively active individuals and individuals that were engaged in brood care. Here are the paper wasp data before, and there was a statistically significant overlap of these two gene lists, suggesting something special about this care gene list with respect to primitive eusociality in the bumblebees and the paper wasps. So we have then some support based on transcriptome analysis, based on gene expression analysis, for the idea that maternal care is unlinked, and we see unlinked gene expression. And we're not actually seeing unlinked gene expression, we're seeing differential gene expression that we are inferring as unlinked gene expression. So this gives us kind of an overall picture, but what might be some of the genes that are involved in the evolution of eusociality? Can we pick up signatures of selection using classical techniques, looking for changes in substitution of the sequence that lead to non-conservative changes, that is, changes that would change the amino acid, that would be assumed to change the amino acid structure and therefore the function of a particular protein? So to do this, we extended the transcriptome analysis, so we created databases, transcriptome databases for 12 bee species, and we have the species arrayed here. We removed a few from the analysis because they were too distantly related. We collected them for some different purposes. The main point is that there are three different categories. We have the green individuals, the species, these are non-eusocial. We have the blue that are primitively eusocial, and the red that are highly eusocial. Here are the phylogenies. Insects are old, for those of you that don't work with insects, so we're talking about, here's evolutionary distance of 80, 125 million years, so a lot of distance there. You see some of the attributes. I'm not going to dwell on all the attributes. The main point is that we had three different groups of species: highly eusocial, primitively eusocial, and non-eusocial. We had about 100 million base pairs of transcriptome data, and then we performed molecular evolution analysis. For those of you specialists in this area, we use PAML with appropriate controls. I'll be happy to talk about that later, to find genes that have been showing signs of positive selection. So did we find genes? Yes, we did. When we compare eusocial versus all the non-eusocial, there were 212 genes showing signatures of selection. There were also genes showing signatures of selection in the non-eusocial species, as we would expect. They're not chopped liver. They've had an evolutionary history themselves. And then when we compare highly eusocial compared to primitively eusocial on the right, we also see a set of genes that show signatures of selection. Who are these genes here? Just a couple of genes, and then I'll give you an overall picture. One that we're particularly interested in is DUNS. DUNS has an amazing pedigree in studies of neuroplasticity and learning and memory in Drosophila, and so finding such a gene showing signs of selection is provocative. That's all that we can really say about it. We don't know anything in particular, how it's been changed, what might result in such a change, but it's an interesting gene to have been identified. Chromatin remodeling, well, that's intriguing because that sort of raises the possibility of wholesale changes in gene expression, the availability of genes to be turned on and turned off by transcription factors. So that's the kind of change that could give rise to massive changes in gene expression. Here are the overall trends. So the way this slide was derived is we take the genes that have been identified as showing signatures of selection, some 200 genes if you'll recall the pie chart, and then take the level of analysis one level up in refinement. Rather than look at individual genes and point and gawk at them like I just did, we look at gene ontology analysis, which identifies biological themes. So it's an ontology of annotation based on the model genetic organisms such as fruit flies and so forth that assign functions, molecular function and biological process annotation to the genes. And so you can feed them in and ask not just, oh, is there a couple of genes showing something, but are there biological themes that are statistically enriched in this rigorous way? And so here are then these results. So when we compare the genes that show signatures of selection, when we look at all eusocial versus the non-eusocial, we get a hit of signal transduction, gland development. We also see these traits right over here, a lot of metabolism. And then when we compare highly eusocial and primitively eusocial, again, a lot of metabolism. That, I might say, would be sort of the dominant theme of our results in terms of signature of selection. I'm just going to close this door here. I'm coming back. So, so we see this. We see some strong hits primarily having to do with metabolism. Okay, so that gives us an initial toehold into areas related to the evolution of eusociality. Obviously, there's a lot to follow up on in terms of the signatures of selection. We badly need to look at some of those genes and then look at the proteins, predicted protein structure, to begin to really extend that to see what kind of changes might be occurring. So this is just the initial approach to this. In terms then of the evolution of social behavior, looking at division of labor, let me begin with this general slide. What it appears as though hunger and reward pathways have been co-opted during social evolution. And I'll show you some data to support this. It looks like foragers go out, and the forager brain gene expression profile looks like it's a hungry bee. But what's curious is that when honeybees go out, they're not hungry. They eat before they go out. There's food in the hive. They ingest the food. It's their fuel. They use it, and they go out and collect food and bring it back. But some of the basic hunger pathways are engaged in the foraging process in ways that suggest this co-opting kind of theme. So let me just give you the background into behavioral maturation. Adult worker honeybees live about 6 weeks. They spend the first 2.5 to 3 weeks working in the hive, engaged in such tasks as brood care, which we've spoken about in a different context. We call those bees nurse bees. Then bees move into middle age about the 2nd week of their life. Happens fast if you're a bee, happens fast if you're a human. And then at 2.5 to 3 weeks of age, they make the final transition where they become a forager. They collect nectar and pollen. You'll notice a compass there. Prior to the onset of foraging, bees take orientation flights. They learn the direction that the sun travels. They learn the position of prominent landmarks in the environment. They use that information to forage efficiently and also to structure their dance language, which we'll talk about in a couple of minutes. And then just prior to the start of foraging, you also have defender bees, bees that work as guards at the entrance, and we'll be talking about them towards the end at the end of the talk. So that's the basic pattern of behavioral maturation. It sounds very stereotyped, and those of you who don't work with insects might think right away that, yes, that fits with our idea of insects as robots, but it's more complicated than that. It's very flexible. Bees can respond to changing colony conditions. They can speed up, slow down, and even reverse their pattern of maturation in response to colony needs. We have identified several social factors, particularly pheromones, that are involved in triggering those changes, modulating the rate of behavioral maturation, which I won't be talking about today, but it is important to make the point in general that the rate at which bees grow up and switch jobs is socially regulated. So what we see is that there's a big role for nutrition in this process. Bees go on a diet prior to the onset of foraging. They lose about half of their abdominal lipid. This is, again, work from Amy Toth. So here we have foragers in green and the nurses in pink. On the y-axis, the amount of lipid in their fat bodies. And in 2 trials, you can see that foragers are about half the amount of lipid as do nurses. This loss of lipid prior to the onset of foraging is causal. if you treat bees with a substance, which we have, called TOFA, which inhibits fatty acid synthesis, and the validation of that treatment is shown on the bottom here. Again, abdominal lipid. You can see that it is having the predicted effect, lowering the lipid levels. When you do that, you see an early onset of foraging. So the loss of lipid has a causal effect. The nutrition connections related to division of labor go far beyond that. We see evidence for what I call signals crossed. Insulin signaling increases in the brain and the fat body of bees and foragers relative to nurse bees. And that's the exact opposite of what goes on in vertebrates, where high insulin signaling is associated with a high nutritional status situation. In honeybees, it's the opposite. So high insulin signaling is associated with foraging. I just told you that foragers are skinnier. We've also done other more manipulative experiments, manipulating diet and so forth, and that pattern holds, which I won't be showing you today. We also have two hormones that turns out are one hormone and one signaling molecule that are connected to insulin signaling, juvenile hormone and vitellogenin. They play a role in pacing behavioral maturation. And here are some treatment data that support both of these. So on the right, this is the work of Seth Ament, a former graduate student who's now a postdoc at Berkeley. If you treat bees with a substance called rapamycin that down-regulates insulin signaling, we, remember, have high insulin signaling in foragers, so now we're down-regulating it. We predict the delay in the onset of foraging, and that's what we saw. On the y-axis, the number of bees foraging, and on the x-axis, age, and you can see that the onset of foraging is delayed. There are fewer foragers. This is a loss of function, so I need to point out that when we trace these bees, we do see foragers just at a later date. So they're not sitting at home sick. They are just delayed in their foraging. Likewise, a hormone manipulation or a molecular manipulation that addresses the juvenile hormone story is on the left side. So when we treat bees with RNAi interference, which downregulates the expression of USP, ultraspirical, which is a transcription factor that's involved in mediating the effects of juvenile hormone, again, we see a delay in the onset of foraging. I'll just point out that this treatment, this RNAi treatment, was done in fat, and I'm emphasizing that because we're going to be talking about connections between fat body and brain in communication and structuring and regulating behavior. So in general, we see these different, atypical nutrition behavior connections with respect to insulin signaling, juvenile hormone, and I didn't mention vitellogenin, a signaling molecule, a lipoprotein involved in a variety of functions that Gru Amdam's lab, who was a postdoc here at UC Davis when Rob Page was here, has gone on beautifully to show novel functions involved in division of labor as well. So USP as a target, as a mediator of juvenile hormone activity, as I just showed you, then becomes an attractive player in the system that we decided to work with further to flesh out the roles of nutrition and insulin signaling and hormone signaling in the division of labor story. So this picture here, this figure, summarizes some work that we did in collaboration with Ripon Pali's lab at University of Kentucky, which basically identified a JH response element that allowed us to clearly point out that USP is involved in the regulatory apparatus for mediating juvenile hormone responses. So USP here in yellow is one transcription factor that collaborates with other transcription factors to mediate the juvenile hormone response. So showing that then told us that USP would be a good target to work on to further elaborate the signaling mechanisms associated with this process. So we then engaged in an experiment. First, we developed an antibody to USP to be able to do chromatin immunoprecipitation, which is a method that takes advantage of antibodies to find the proteins that are talking to USP. So USP is a transcription factor, binds in the regulatory regions of genes to turn them on, to turn them off. Who are those genes that it turns on and turns off? Chromatin immunoprecipitation is one method that can give you information on that. So then you use chromatin immunoprecipitation. We followed that with a microarray analysis colloquially known as chip-chip, to identify those genes that are bound by the antibody to USP. And here is the overall results on the right. So we found lots of genes that are putative targets of USP, 1,300 binding sites. Now, some of these are going to be false positives. These are not validated experimentally using the tools of Drosophila genetics. These are based on the binding of the antibody. These 1,300 binding sites putatively represent about 800 genes. We see 50% overlap with the targets that have been identified in Drosophila. So that's encouraging. We see equivalency in the binding sites between nurse and forager, which is interesting in the sense that we might have imagined a simplistic model where USP regulates different genes in the nurse fat body and in the forager fat body. Did I say this was in fat body? If not, I forgot, and let me point that out. This was a fat body experiment. We might have expected different genes. We don't see that. And so instead, then, what we think might be the case is that USP may be involved with different transcription factors in these two different contexts of nursing and foraging. We've not proven that. That's just a speculation. So here are some of the genes, and I'm highlighting ones that make sense and that are intriguing. So we have some genes involved in juvenile hormone signaling, as we might expect, some genes involved in USP signaling. We see USP itself, interesting suggestion of self-regulation. Some nuclear hormone receptors that have known to be involved in hormone signaling, suggestion of methylation. DNMT3 is involved in de novo methylation, a major epigenetic factor. And then another gene that was encouraging, the foraging gene. In other studies, we've shown that the foraging gene is a gene that involved in regulating behavioral maturation. It's upregulated in the brains of foragers. When you treat bees with a method of upregulating that system, then you get early foraging. So some genes that are really making a lot of sense. Many of the genes have been validated, not in a wet lab experimental sense, but they've been validated in a dry lab informatics sense, and that's this right column of predicted USP binding sites. So you have the chip targets based on the chip data here, and then bioinformatically you can ask, do the promoter regions of those genes that are identified by the chip method, do they have the cis-regulatory element that would be binding USP? And so many of them do, as you see here, and this is how we would depict that graphically. So indicating the chip-chip sites in pink, and then the computationally predicted USP binding site. So not every one of these genes is going to turn out to be a USP target, as I said, but a lot of them are based on the confluence of prior data from Drosophila, the logic of some of these, and the fact that we have bioinformatic confirmation for some of them. So USP then giving us a nice set of genes to begin to elaborate the network that's involved. One other piece along the lines that relate to combinatorial control, we find other cis-regulatory motifs that are enriched in these USP-bound regions, supporting the idea that these cis-regulatory motifs, of course representing transcription factors, other transcription factors, then could be working together with USP. I circled two of them. ADF1 is one that we have our eyes on because it shows up in every one of our cis-regulatory scans that we perform. That is, we get a gene list from a microarray experiment comparing behavior state X and Y in honeybees, socially regulated and so forth. We scan the promoter regions for known motifs, Drosophila-based characterized motifs, and ADF1 comes up a lot in our socially regulated gene list. FTZF1 is another one we have our eye on because in collaboration with Nathan Price's lab, a systems biologist, we have created a brain transcriptional regulatory network based on a large study of many different behavioral states, which I won't be talking about, other than to say that it was a large study, involves 853 individual brain profiles, 48 different behavioral states, and this is a graphical depiction of the brain transcriptional regulatory network. What we find is a set of global transcription factors, just 4 of them, that are active across all of the different states, foraging, maturation, and aggression. That is, these 48 behavioral states can fit into one of these three categories. We did a bunch of different experiments that capture different aspects of foraging, a bunch of experiments that capture aggression, a bunch of experiments that capture maturation. Some global regulators active in all three of those contexts, and then some behavior-specific transcription factors that are indicated in these squares here that represent transcription factors active in one set of one context and not the others. So the model was encouragingly robust. We were able to predict about the expression of 25% of the genes based on the expression of the transcription factors, and the predictive value of those genes was very high. The correlation was 0.87 predicted versus actual gene expression data. So with this network, and looking at the previous slide of the chip-chip data, you remember FTZF1, you remember USP. So here's one of the sub-networks in that larger network. FTZF1 is playing a prominent role. It is a juvenile hormone-regulated nuclear hormone receptor in the bee brain. We've done this work with Susan Farbach. We see it's predicted to regulate a bunch of genes. And then the most important point I'm trying to make is the third line there. 6 of the 13 predicted transcription factor targets of FTZF1, so sort of the middlemen, the intermediate level, below the master regulators, but above the non-transcription factor genes, are also predicted targets of USP. So we have bioinformatic evidence of the cis-regulatory elements for FTZF1 in the USP promoters, and then we have these data here. And then the categories, again, using gene ontology, among these 146 genes, vitellogenin signaling, lipid metabolism, neural development, and behavior. By now, those should be familiar sounding categories. We keep hitting those from these very different experimental approaches. So to summarize this part of the talk, division of labor among workers seems to involve evolutionary novel relationships between nutrition and hormone signaling pathways. So we've been talking about nutrition, we've been talking about metabolism, but let's get down to brass tacks here. Let's talk about what's really important, sweets. So, of course, we have no problem imagining this figure here. What about this one? Is it possible to interpret the way bee behavior works with this kind of model? So bear with me and then see what you think when we get to the end of this little story here. So the background has to do with the dance language. Dance language, of course, symbolic language, communicates distance, direction, and the value of the food. The bee, if the bee is successful, that is, if she finds food, she will come back to the hive. And if the hive gives feedback to the bee indicating that it's hungry, that it wants the food, then the bee will proceed with dancing. I belabor this point to make the following point. Dance is not a reflexive behavior, it's a modulated behavior. The bee assesses the quality of the food it's taking, it assesses the distance, it assesses how hard it is to get the food, it assesses the colony's need for the food, and then it, quote, makes a decision to either dance or not dance. Andy Barron, a former postdoc in the lab who now has his own lab in Macquarie University in Sydney, Australia, did some work to show that octopamine causes bees to dance more. That is, if they go out, they find good food, and if they're treated with octopamine, they are more likely to dance than the control bees. We were very enthused about those results because octopamine is known to be involved. Octopamine, by the way, is a biogenic amine, neuromodulator involved in regulating a variety of functions in insects, one prominent one being eating behavior. Octopamine makes insects eat more. A famous experiment that was performed by Vince Detier, you treat bees, like treat flies with octopamine, and you can get them to eat so much that they explode. So it's a great undergraduate lab, but don't try it at home. So octopamine makes insects eat more. We know the natural history of honeybees. When they find good food on flowers, they don't eat more. They come back home and dance more. So there's kind of the beginnings of this me to we sort of thing where you take the selfish behavior and you turn it into social behavior. Could octopamine be mediating that me to we evolutionary transition, which may not be a driving force in the evolution of eusociality, but certainly might be part of the whole process? Cooperation is required in a eusocial context. So the octopamine data fit all that. The only problem with that scenario is that the reward system in insects is not well studied. And many forms of insect behavior, one could imagine a simpler explanation than having to invoke the reward system. By contrast, in mammals, certainly in us, we have no problem invoking the reward system for anything. I like this, I don't like this. For insects, it's a little more of a gray zone. I would argue that dance language is clearly one of those behaviors that you can't use a simpler explanation like the whole idea of stimulus response. But still, we were on shaky ground. So the simple solution is to roll up one's sleeves, map out the reward system very carefully in honeybees. It's not been done before. It's been done a little bit in Drosophila, but still, even the reward system is not really well mapped in Drosophila either, certainly in the context of these kinds of rewards, and then really flesh it out. But I thought before we do that, let's take a shortcut and see if we're on the right track. And so we took advantage of a little known fact, which is that cocaine exists in nature as an insecticide. That's what it's there for. It is pharmacologically characterized, it was some 25 years ago, as an octopamine reuptake inhibitor. So there's a transporter, octopamine transporter, that is involved in sucking up the octopamine after it's released to modulate the activity of octopamine. And what cocaine does is it inhibits the action of that reuptake mechanism, swamps the synapses with octopamine, and then it has then a debilitating, paralyzing effect on insects. So it's a natural insecticide. The reason why you know it perhaps better is because dopamine and octopamine are so chemically similar, it also acts that way on humans to potentiate the effects of dopamine, which of course in shorthand is the brain's pleasure juice, and so the rest, as they say, is history. So we thought we would take advantage of this. If you've just been listening to what I said, pharmacologically it was a trivial experiment. Does octopamine make bees dance more? Yes. Should cocaine make bees dance more? Yes. But, you know, stranger things have happened in science, so we thought we would do the experiment, and if we got the predicted results, we would know we're on the right track of interpreting all this within a kind of a reward framework. Whereas if we get, you know, kicked in the teeth, then we would say, well, maybe we have to rethink it. So we did the experiment, Andy did the experiment. Just parenthetically, I want to show you one of the methods that we use to perform this experiment because it's important for appreciating the results. It allows me to show off cool methods with bees that I can't take any credit for, so that's the best form of showing off. This is a tunnel that was developed by Manyan Srinivasan in Australia to manipulate the perception of bees' distance. And he did that in order to show that the way bees measure distance is by optic flow, the amount of stuff that flows past their eyes. And so he showed that if you train bees to fly through this tunnel that you see on the right, and if the pattern of checkerboards is really, really sparse, so that very few images flow past the retina, when they get to the end of the tunnel, they go, that was a really short distance. And they tell you that because they dance the round dance, which indicates a short distance. If you, same tunnel, same bees, same chamber, same colony, blah, 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 and you just change the walls of the tunnel so that you have a very dense checkerboard pattern like this, they get to the end of the tunnel and they go, wow, that was a long distance. And they tell you that by dancing the waggle dance. So that's important because we did all this work in a flight enclosure, which is about 25 meters long, and we had to do that in order to control all the ambient environmental stimuli and train bees to a given feeder to be able to ask, do bees that go to this feeder, does our cocaine treatment increase the likelihood of dancing? If we didn't control where they go, you know, there'd be too many variables. So this is a way to control where they go by having them in a confined quarters. But this distance is too short to do waggle dance. They would only do round dances under natural conditions. What's wrong with that? Well, what I mentioned before is that octopamine has effects on motor activity as well as reward, just like dopamine has those effects in humans, right? Parkinson's is a disease of the motor system, just like dopamine is considered the brain's pleasure juice. So the worry was that if we get an effect of cocaine on dance, it could be interpreted, well, maybe it's just causing a lot of wiggly motions because it's having motor effects, and so it wouldn't be definitive on dance. So we used this technique to get good waggle dances. So all our dances were video recorded. We could show that the dance, the quality of the dance was not distorted or degraded in any way at all. The only thing that changed was the likelihood that the bees would dance. So here are the data. So we have octopamine was our positive control, the pink bar. On the y-axis, we have the likelihood of dancing. We titrated the situation so that in our control bees, the likelihood of dancing was pretty low. We can manipulate that. So we made it sort of a marginal feeding resource that not too many bees liked enough to dance, to obviously to give us room to see the treatment effect. So we have two controls, bees untreated, bees treated with the solvent alone, DMF, and then the octopamine treatment you can see caused an increased likelihood of dancing. The cocaine caused a similar increased likelihood of dancing. That's the orange bar. And then pharmacologically, we treated also the bees with an octopamine antagonist along with the cocaine, and that blocked the cocaine effect, which is, gives us one line of evidence supporting the idea that this was affecting the octopamine system. So cocaine makes bees dance more. We've gone on to show two of the gold standards associated with addiction. You can get condition-place preference and withdrawal in honeybees, which supports the idea that cocaine's effects are highly conserved across mammals and insects. That is, we're looking, we think, at the profile of addicted bees. Cocaine can addict bees. And then this supports the idea of a me to we shaping of the reward system. So there's a lot of things we don't know. Do bees dance because cocaine makes them like their food more? Is that what's going on? Does it increase their valuation of the resource? Or does cocaine enhance their, quote, pleasure from dancing, doing a good deed? We're not sure about those, but those sort of animate the kinds of questions that we hope to address in a comparative approach, where we take this schematic, the idea that we're looking at the hijacking of the reward system in evolutionary time. If we compare solitary bees to social bees and apply certain stimuli, drugs such as cocaine, food, rewarding stimuli, and then monitor the brain's response transcriptomically, changes in gene expression, and then do network analyses to identify modules that show differential response, perhaps in a solitary context compared to a social context, to begin to get at what kinds of changes are going on in the brain during social evolution related to the changes in reward. Okay, so then the last part of the talk then deals with, you've got a division of labor system, and how might this be fine-tuned? We chose to address this question by looking at aggression in honeybees, because it's a very rich system. So honeybees employ aggression to defend their hive. This, by the way, is my Patty Hearst slide. Please chuckle if you're old enough to understand. Thank you. The numbers get fewer each time I give the talk. So, so bees employ aggression defensively to defend the hive. They amass huge amounts of resources. They turn nectar into honey. There may be 50, 60, 100 pounds of honey in a beehive at any one time, maybe 10 pounds of pollen, maybe 5 pounds of brood. If you're Winnie the Pooh and you want a meal, would you go to individual flowers and suck up 1 microliter of nectar, or would you go one-stop shopping to a beehive? So their success has made them vulnerable. They have evolved then very impressive defensive systems. And these defensive systems I will describe for you in a moment. They involve behavior, they involve chemistry, they involve social organization. The general question that we're asking is then, do differences in behavior evolve via changes in gene expression? So we have a very nice comparison that indicates that changes in aggression have occurred as honeybees have spread through the world. So African bees show increased aggression, and this we presume is due to natural selection. European strains, subspecies of bees, show decreased aggression. We assume this is due, at least in part, to artificial selection by humans. So we see changes in how aggressive a colony is in response to a disturbance. This can be measured by the number of bees that appear at the entrance. So this is a regular man-made hive. This is the entrance to the hive, and you can disturb the hive scientifically by kicking it, and then measure the number of bees that come out, or you can put a leather strip in front of the hive and get stings and count the number of stings. And when you do that, you can quantify and show strains that are more aggressive and less aggressive. And so we use this system to address this question having to do with, might we be able to get the sense of an evolutionary change that relates to changes in gene expression? So when we think about that, you think about coding region changes that give rise to protein, regulatory region changes, and of course, novel genes. We're inferring then, if we're looking at changes in gene expression, regulatory region changes would be going on. Sort of another piece of background information is the notion that with the advent of genomics and the ability to do gene expression profiling, we now have what I call the first phenotype, the place where the genome meets the environment, and that's in measures of gene expression. So genomics and the first phenotype reveals that nature and nurture both act on the genome. So you have hereditary effects on the genome and gene expression. You have environmental effects on the genome via changes in gene expression. And the key factor is to put this in the context of differences in time scale. So evolutionary differences relate to hereditary differences over a long time scale, an evolutionary time scale, and then environmental differences relate to, of course, a very short time scale. So that's sort of the background context to set this study up. The system in honeybees is very rich, and it allows for this kind of dissection. So there are hereditary differences or hereditary components to aggression. I already talked about those, African versus European. There are behavioral components. There are groups of bees that specialize on defensive behavior. So there are bees that patrol the entrance, that show a characteristic alert, if you'll anthropomorphize, indulge me, by anthropomorphizing. Alert posture, their antennae are forward, their wings are partially back, their forelegs are off the substrate. Long-term behavioral observations have revealed that these are guard bees. They respond first to the intruders. There's another group of bees called soldiers that the guard bees release alarm pheromone. Soldiers are the first group of bees to respond to the alarm pheromone by flying out and looking for something to sting. So there's a behavioral component. It's organized socially, as I just said, by the release of alarm chemicals. And then there's a maturational component. Old bees are more easily aroused than are young bees. So former postdoc in the lab, Cedric Alou, who's now on the staff at INRA in Avignon, France, did 3 different microarray experiments comparing those contexts. So aggression over 3 time scales, if you were. He compared African versus European bees, both guards, soldiers, and foragers as kind of a negative control. He compared bees, the effect of alarm pheromone on bees, on European bees, and compared old and young bees, all brain gene expression profile. This is just a summary. So what you see here are the numbers of genes showing differential expression. And so guards and soldiers showed greater differences in expression when we compare African and European bees than did foragers. So about 250 genes different here, over 500 and only about 60. That was an encouraging result because there are more differences, more known differences in aggression between African and European bees than there are for foraging. So while we can't say that all of these 249 or 538 are related to aggression, because there are lots of other differences between various subspecies of honeybees, but we can say that the dominant trend is consistent with the idea that that is one major difference, and so therefore differences in brain gene expression should be heavily represented there. So alarm pheromone, when you expose bees to alarm pheromone, you get a difference in gene expression. 437 genes showed differential expression. Just as an aside, that's interesting because alarm pheromone induces an immediate behavioral response. You put alarm pheromone at the entrance, and as Norm Gary or Robin Thorpe can tell you, you'll get an immediate response at the entrance. Gene expression responses are not immediate. They are of a time course measured in terms of minutes, up to half an hour, up to even several hours. So what we see is alarm pheromone induces an immediate behavioral response, which is not genomic, and then a genomic response, which we've shown in studies that I'm not going to show you, is at least correlated with an increase in vigilance or alertness of a colony over periods of hours after exposure to alarm pheromone. And then finally, to fill out the top, in green, differences between young and old brain gene expression, and many genes showing differences there. But the point of this is to show the convergence, and that is there's a statistically significant overlap of those genes. So there are genes that converge and show differential expression in all 3 of those aggression comparisons. African versus European, alarm pheromone, or old versus young. And then again, when we take the level of refinement up one step and look at gene ontology, there are a number of gene ontology categories that show consistent and coherent response. And I'm highlighting for you, once again, metabolism. So the interesting thing about metabolism here is not just that it's coming up yet again in this part of the talk as it has in every other part of the talk, but the aggressive state was associated with downregulation of aggression. And that was such a counterintuitive result, we, for the first time in this talk, extended the results beyond just mRNA and took it to the level of protein. So collaborating with Julie George, who studies Parkinson's and mitochondrial diseases of the mitochondria, neuro diseases of the mitochondria, we made brain mitochondrial preps and then looked at the enzymatic activity in particular complexes, complex 1, 4, and 5, that are predicted by the gene expression differences. So one of the nice things about working with changes in metabolism is that your annotation, that is your function of the genes, doesn't just say metabolism, it says complex 1 metabolism, complex 4, complex 5. Why? Because biochemists have figured out metabolism before genomics came on the scene. There's exquisite detail. So we could use specific substrates to be able to test for the enzymatic activity that are predicted. And we find the exact same pattern that I showed you for the mRNA. That is, there's a downregulation of brain metabolism, now seen at the level of enzyme activity, respiratory enzyme activity in complex 1, 4, and 5. That is, killer bees, African bees, lower levels compared to European, old bees, lower levels compared to young, and alarm pheromone treated bees, lower levels than control bees. So very interesting pattern, raising interesting questions about what's going on in the brain with respect to overall metabolic activity. These are brain data, mind you, what's going on there. For the purpose of the talk, just sort of general hit again on metabolism. This theme of gene regulation across 3 timescales is strengthened by, again, a cis-regulatory analysis. So we take those gene lists that I just were showing you different aspects of a moment ago, and again, search the promoters of those genes for cis-regulatory elements, looking for statistical enrichment of particular motifs that represent particular transcription factors, and these are the best hits. First of all, the result is that we see some that are statistically enriched, again, across all 3 timescales in the gene lists for all 3 timescales. The ones in yellow, DF1 has been implicated in aggression. It's a transcription factor implicated in aggression in vertebrates. Hairy, a well-characterized Drosophila, quote, developmental transcription factor that, again, we find coming up on many of our social regulated gene lists, not just the ones here. And then I don't have time to show you some data that are suggestive of epigenetic changes going on as well that are consistent across the 3 timescales. So to summarize the last part of the talk, this new nature-nurture perspective having to do with the first phenotype provides a framework to study the relationship between gene regulation and behavioral evolution. The genome seems to be very sensitive to social influence, that alarm pheromone response. Kind of summarizing these results, the killer bee molecular profile, when you kind of compare the gene expression pattern of African bees to the expression pattern induced by alarm pheromone, the African bees look like they're, quote, on alarm pheromone even when they haven't been exposed to that. And there's some interesting old genetic theories having to do with genetic assimilation that I'd be happy to talk about that these results kind of call to mind. And then finally, a purely mechanistic insight, downregulation of brain metabolism is associated with behavioral arousal. So to summarize, we've talked about some studies related to the evolution of eusociality, the evolution of social behavior as evidenced by division of labor, and then the adaptation of social behavior having to do with changes or tuning in aggression. We have implicated by means of evolutionary transcriptomic analysis, by means of signatures of selection, by means of molecular systems biology, a little bit of physiology, the patterns or the processes that are highlighted in yellow: feeding, nutrition, metabolism, and reward. Going forward, some of the challenges that we face that we're very interested in trying to address, we need to really strengthen our genotype to phenotype matching. So for example, the genes that we found, these provocative signatures of selection for. We need to know more about them, what protein, what aspects of protein function they may have changed, how might those proteins be involved in anything related to eusociality. So we have this hit of metabolism. We can see how metabolism plays out and nutrition plays out in division of labor, but we really need to strengthen those connections. I've emphasized today that social evolution can proceed via changes in gene expression and gene sequence. What about novel genes? Every genome sequencing project comes up with, quote, novel genes. Many of them are just genes that haven't been annotated yet. We don't have enough genomes to provide complete annotation. But there is a general feeling that there may be lineage-specific genes, and we've not addressed those. And of course, this experiment, this scientific paradigm, this approach that we're using isn't well suited for novel genes. We need more discovery-oriented approaches to be able to work in a forward direction, and we don't have those in honeybees. So we're kind of caught in that kind of tautology where we can make hay looking at genes that have known function, but we can't lose sight of the fact that we also need to be able to reach out somehow and get at new genes. Some of the results that we're getting give rise to the notion that perhaps we're looking at a genetic toolkit for sociality, ideas related to the co-option of the reward system, scaling across vertebrates and the social insects. Maybe there's some deep conservation, even though the actual social phenotypes obviously are not considered. Remember the dig I got in about the marine flatworm, so they can't be truly conserved in the traditional sense, but there may be toolkits there that are used, that are called upon to elaborate social life in a repeated way and qualifying therefore as a toolkit. That's just a question. And finally, when we think about the evolution of insect life, and as we get a better handle on some of the genes that are involved and some of the biological processes that are involved, I think it'll be interesting to contrast human social evolution with insect social evolution. Insect social evolution is all about me to we. The individuals have actually become more dependent upon each other. So they have achieved the pinnacles of social evolution, language, warfare, agriculture, as I said, but they've done it by investing in the group rather than the individual. The extreme forms of division of labor render individuals incapable of living alone. Meanwhile, on the other side of the phylogenetic tree, the way that humans have achieved those pinnacles is by an evolution from me to me, 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 me. An increase in the capabilities of individuals. So obviously I'm just talking about the fact that the dominant paradigm in primate and human evolution is the big brain hypothesis, the expansion of the cerebral cortex adding increased capability. So I think it's interesting to frame that contrast of a trajectory that leads to increased capabilities of individuals and a trajectory that largely has led to increased interdependence of individuals. And will we be able to ever flesh that out at the genomic level? I don't know, but I think it's an interesting framework to use, or at least to keep in mind. So let me close by acknowledging some of the people whose work I spoke about. Cedric Alou on the aggression work, Seth Ament on the nutrition work, Andy Barron on the dance work, Brielle Fishman and Hollis Woodard on the molecular signatures of selection work, Christina Grosinger on the nurse forager work, Charlie Whitfield as well, Amy Toth on the nutrition work, Ying Wang on the USP, chip work and the network analyses, my lab manager, Tom Newman, B-Lab manager, former B-Lab manager, Karen Pruitt, several people we've been collaborating with on some of this work, Andy Clark on the molecular evolution work, Matt Hudson on the gene network analysis, Richard Molesca on the epigenetics, Craig Mizzen on the USP, chip work, Nathan Price on the brain transcriptional regulatory network work, and Saurabh Sinha on the cis regulatory analysis work. Thank you very much. Andy spent too long on the introduction. It's not my fault, but I'll be happy to take any questions you have.
What was the source of your African bees? Were they American African bees, or were they African bees?
The source of the African bees were in Mexico. They were behaviorally phenotyped to show the normal level of aggression.
I asked because your nature-nurture paradigm may be even more subtle than you suggest. Because my experience and the experience of a whole lot of others is that if you look at African bees in Africa, they're no more aggressive than American bees. Otherwise, I wouldn't be here, I guarantee you.
Was that Apis mellifera scutellata?
I couldn't tell you. I mean, there's several places where I've experienced this: Kenya, South Africa, and Cameroon.
Because there's a lot of variation in the subspecies in Africa.
I wouldn't be
And
surprised.
it was the 26 queens that were introduced, or 52, depending on what story you believe, came from an apiary that was known to have very aggressive bees. There are other subspecies in Africa, such as in Kenya, Apis mellifera monticola, that are very gentle. So these, I think,
We call Cameroonian bees gentle, but they certainly weren't all that aggressive.
Yes.
So what's the pattern of insulin signaling in the brain of the bees where you see the repressed metabolism during arousal? Have you looked at that?
In the aggression kind, we have not looked at that. We've not looked at that. And some of those genes are expressed at a low level. So microarray data, I could say, well, we can just go look at our microarray data and see. They're not that reliable there. We've had to use qPCR for the insulin signaling genes. So I don't have an answer to that. But I guess inferring from that, foragers also have low levels of brain metabolism, and they have high levels, those were the data I showed you, high levels of insulin signaling expression in the brain. So I might extrapolate and say that's probably what we would see. Is that interesting, that they're opposite? Yes. Why?
Oh, I mean, I work on insulin signaling, and we're very interested in the connection between immunity and metabolism at that level. So.
I'd like to hear more about it afterwards, maybe. Thank you. Yes?
Have you looked at the effects of octopamine on non-social outgroups or lineages that may have lost eusociality?
So that's the plan. That schematic that I showed about the evolutionary hijacking of the brain. So we want to treat, right now we're sequencing genomes to have the wherewithal to do the transcriptomic analysis. But we want to then expose the solitary and primitively eusocial and highly eusocial, two independent replicates, if you will, of each of those to natural rewards, to drugs, and to octopamine, and then do the brain transcriptomics to see if there's what kind of differences there might be. So we've not done that yet. Any?
It's along that vein. Have you or anyone else working on termites or the somewhat social aphids or any of these other groups in terms of what they're doing genetically?
Not yet. There is a termite project that was approved. So Mike Scharf at University of Florida is moving to Purdue, is heading up a termite genome project. And so, soon there will be that. I'm not aware of any P. aphid was done, but that's not a eusocial species. P. aphid was sequenced. Was it published yet? I can't remember. But that was done. And as I told a few people today, I've started an initiative to try to get funding for 5,000 insect species, genome sequences of 5,000, and then we won't have to ask these nickel and dime questions anymore. This one, yes, that one, no. We'll have them all.
So, along the same lines, I guess, I'm intrigued by your idea of the toolkit, which, you're saying spans, a big chunk of benzoin life, right? So, would the prediction be, and is there anything you can do with this, that in sort of somewhat social systems, or something, that you would always repeatedly see sections of that toolkit that would show up over and over again in truly distantly related to these organisms, not just with the social insects or something.
That's the prediction. And so first of all, say it's the toolkit idea comes from EvoDevo, and the data there are Hox genes that are involved in patterning very disparate forms of embryonic and metazoan development. And so it's more problematic to apply that idea to social behavior because you're dealing probably with neural substrates that are not conserved in any way at all. Although there's some recent data from basal vertebrates that are suggesting otherwise. But so it's it's interesting. But the simple articulation of the idea is that there may be elements, that there are elements of signaling pathways, of transcriptional regulatory networks that are, I sort of call it, it's a culturally biased analogy, but the Ghostbuster metaphor. You know, have you all seen the movie Ghostbuster? You need plasticity, who are you going to call? So that sort of thing. And there are a couple of intriguing examples of that so far, not yet across the broad span of evolution that we're talking about, but FOXP2 gene involved in speech and communication in many different vertebrate species in cases where the communication systems are thought to have evolved independently, involving even different sensory modalities. And then the foraging gene in invertebrates, in C. elegans, and honeybees, and Drosophila, and in ants, and now some suggested data from Marla Sokolowski's lab in humans. And so there are a couple of these genes that are showing these interesting pedigrees, and are they sort of the tip of the iceberg for this gene toolkit, genetic toolkit idea? That's the premise that's motivating it. Yeah.
So with regard to your very last bullet point about interdependence and me, I was wondering if, like, where the evidence for that comes from. Like, for humans, like, not only, so there's lists of collaborators, I have collaborators all over the world. I had collaborators make my lunch. I couldn't do it myself. Plus there's theories that say that human sociality is largely based on, you know, shared care. Yeah. On the honeybee side, they seem to have bigger brains than other members of their group. They have stronger defenses in some cases in terms of stingers. If you like asked a honeybee, like, how altruistic are you? Would they give are you saying that they'd give a different answer from like someone who made my lunch, or like, is there a scientific basis for that, or?
Right. No, that's an interesting question. No, not much of one, except to say that what I'm talking about is at the root level. So in primate and human evolution, there has been an increase, a dramatic increase in the capabilities of the individual. And in social insects, there are a couple of cases of dramatically new abilities. Agriculture, although if you look at the agriculture, each individual is doing very, very small amount of things. The brain stuff in social insects is not holding. There is an increase in brain size associated with a variety of different ecological contexts. Work of Sarah Ferris on this, in generalists and specialists, for example. So it's no longer, I know the earlier literature you're referring to, and you're accurately representing that, but it's no longer, I think, the case that is true. So it's an idea, it's a heuristic, and it's more along the lines of there's much bigger increase in capability if you look at primates to humans than if you look at a solitary bee or a digger wasp relative to a social insect. Yes.
So this is probably somewhat related, but in honeybees, is there any evidence of synapse pruning happening during maturation?
What we see, so we've studied this with Susan Farbach at the level of volume changes. So the volume of the mushroom body, the insect region associated with learning and memory, increases as the bee matures and is largest in foragers. That's involved at the cellular level, are based on, at the cellular level, on an increased dendritic arborization. So we have not, though, seen a synaptic pruning scenario, but we've not taken it down to the level of Golgi analysis that would be required to really see that. But we do see increased dendritic arborization, which would suggest an actual increase in synaptic connection, as you know. So we don't see the pruning sort of thing.
Your bullet about me to me, and me to me to me, and me to me, it got me thinking about whether there's an insect equivalent to philanthropy. Is there any cases, you know, where insects become so successful as individuals that they give back to the greater good?
Yeah. I don't want to tell Ken he's going to steal my donor, so not aware of anything
So that's uniquely
like that. Yeah, interesting. Thank you very much.
All right.